This figure, we have shown you before, explains the differences between the eukaryotic genome and prokaryotic genome. Because the eukaryotic genome has many copies of similar genes and repeat sequences, the long-read sequencer, such as PacBio, has more advantages than the other platforms. Here is the amount of sequencing data obtained from this analysis. The long reads yielded 345.5 gigabases on 28 cells of PacBio sequel, of which we used 120.7 gigabases of reads with over 35 kilobases as long. This amount is equivalent to 49.5 times of the domestic cat genome. The short reads were obtained by Illumina HiSeq X, which yielded 1.54 gigabases of 151 bases with paired end. We used these reads to assemble contigs and to correct errors. The terms read means the sequence obtained from the high-throughput sequencer, and the word contig means the sequence connected read. The PacBio reads were assembled with the tool named Falcon Unzip. Falcon Unzip is an assembler designed for long read, and it considers haplotypes when connecting the read. Next, we use the tools named Arrow and Pylon. Arrow creates a consensus sequence with long read to correct errors. In contrast, Pylon uses the Illumina short read to correct errors. Next, using HiSeq and optical mapping and reference-guided assembler, we linked the contigs to make the chromosomal level sequence. In the HiSeq method, the DNA that is spatially proximate in the nucleosomes is extracted and read sequenced using the high-throughput sequencer. Then, we can find the region close to each other. The summary of the HiSeq method is shown on this slide. First, we make the cross-linked DNA in the intact chromatin structure with formaldehyde, and then we can cut the cross-linked DNA with the restriction enzyme. Next, we bind biotin to the end of DNA and ligate the DNA again. The last step is extracting the fragment which found biotin to construct for sequence library. When they are read sequences in paired end and mapped to our genome, the chimeric fragments are mapped to different contigs for each pair. Therefore, we can consider that those contigs exist close to each other. In other words, this method is possible to infer connections between contigs. Next, we try to connect the sequences by optical mapping. This method is developed Bionanogenomics company. First, we labeled restriction sites with fluorescent on the genome. Then, we get an optical map by labeling patterns such as the figure at the top right. They were aligned to make a consensus map. Next, we aligned the contigs according to the restriction site on the contigs. The last step in the genome assembly is not shown on this slide. We assembled our genome sequence using a tool named RAGU. RAGU used the current reference sequence for domestic cat named FELCAT9 as a guide to reassemble our genome. This slide summarizes each step of the assembly. Please see them from left to right. To evaluate the quality of our genome, generally we use the N50. N50 means the length of the contigs or scaffold. Starting from the longest contigs or scaffold and adding them to reach 50% of the total length. This value indicates that the genome assembly is more longer and more connected. Please note the terms contig and scaffold means the assembled sequence. Generally, contig means the sequence without the character N, and scaffold means more longer sequence containing the character N. N50 or our genome, which assembled by Falcon and Zip, is 21.3 megabase, but it grew to 149.6 megabase in Haishi by the optical mapping. 37 scaffolds accounted for 99.4% of the total number of bases. N50 was reduced by optical mapping due to the correction of some incorrect assembly. The result of the genome assembly has 2.49 gigabases. N50 is 151.1 megabases. Our genome has 19 scaffolds corresponding to each chromosome, but one scaffold could not be attributed anywhere. To evaluate the completeness of the genome assembly, we also used the tool named BASCO. BASCO has the standard set of genes and evaluates the integrity of the genome assembly. In other words, it estimates that whether the genome assembly satisfied standard orthologs. Ortholog means corresponding genes that have the same ancestor. These standard orthologs are prepared for species. In this case, we use the set for mammals. The data in the squares on the bottom of this slide represent the result of BASCO. The meaning of each is: N means the number of genes exist in a set of mammalian. C means the percentage of complete mapped on orthologs. S means for the ratio of single hit. D means for the ratio of multiple hits. F means the gene found as fragments, and M means for the percentage of genes that do not exist in the genome. in our genome. This means that of the 4,104 genes, 95.6% were found as full-length, 2.4% were fragmented, and 2% were not found.